Oh, I see one of my ex-PhD students has just entered the room all the way from London. So, Iria, welcome. Nice to see you. Enjoy. So, we still have a few people joining. So,
yeah, I see. We're over the 50 mark anyway.
Yeah. Mm. So I think if I just start by welcoming everyone to this Global Food and Environment seminar and just reminding you of the Zoom etiquette. So if you can please keep your cameras and your audio off. There'll be a question and answer session at the end of this session where we'll have about 10 minutes and you can pose questions to our speaker during that time. So if you have any questions, please enter them in the chat and I'll be monitoring that as we go through and I'll pose them to Alan at the end of his talk. So I'm Louise Dye. I'm the lead for diet and health in GFEI, and it's my pleasure to introduce Alan Mackie today, who is our guest speaker. Don't know why the slide's not moving on to you, Alan. Do you want to put yours up?
Yep, there we go.
Right. So Alan is the head of school in food science and nutrition. So he's well known to many of us on this seminar, but he's actually got a background in physics. And he's going to talk to us today about his research, which is really reflecting on his expertise in food colloids. So Alan came to Leeds as a professor in 2016. But prior to this, he spent a long time at the Institute of Food Research. So many of you may know him from there, but I'm sure you're all familiar with his work. And it's particularly exciting stuff around in vitro digestion that we're going to hear about today. So I'll close my microphone and I'll just hand over to you, Alan.
Thank you very much, Louise, for that very kind introduction. The title is very broad, so I apologise for in advance for that, but we're going to talk about different types of structure. So by food structure, this can be obviously natural or artificial. It can describe architecture from the nanoscale, so molecular architecture, through to the centimetre scale, which is kind of bite size. And everything in between. And diet-related health and non-communicable diseases such as diabetes, cardiovascular disease, and some cancers are linked to overconsumption and poor diet, and that links back into the structure of that and the composition of the foods. First of all, I thought it would be helpful to have some definitions. So in the world of digestion, we tend to talk a bit more about bioaccessibility, and the definition of that is the extent to which a compound is released from a food during digestion and becomes available for absorption. So it's potentially available to be taken up and do good. But obviously what we're really interested in from a biological perspective is bioavailability, which is the extent to which a compound can be digested, absorbed and transported to the site of action and actually do something physiological. And bioavailability obviously implies bioaccessible, because if a compound is not bioaccessible, it's never going to be bioavailable, but obviously the reverse is not necessarily true. In addition to that, for any bioactive that's metabolized or cleared from the body, the absorption kinetics, so the rate at which that bioactive compound is absorbed and is reaching the site of action and is maintained at the critical threshold concentration to deliver that bioactive is clearly key. And much research has been done on looking at response to, for example, carbohydrates and proteins in the food arena. Obviously, there's also lots of work being done in the pharmaceutical arena, looking at particular drugs and their uptake kinetics. But in the food arena, as an example, you might think about glycemic index. So we know that a low glycemic index, for example, with a high amount of fiber and minimally processed whole grain products, reduces glycemic and insulinemic responses and lowers risk factors for type 2 diabetes, for example. So there is a link directly between the uptake kinetics and health outcomes. So what are consumers interested in regard to health and food? Very popular, particularly among students, to be interested in protein and associated sport nutrition, and we'll talk a little bit about that. But here's a graph from Google Trends looking at what consumers are interested in with regard to the search term protein. This is just for the UK. And one of the outstanding features you'll notice is that it varies periodically through the year. And this trend has got worse as time has gone on. So now we tend to look for protein immediately after Christmas. So these peaks more or less correspond to the beginning of the year, and that interest gradually wanes as we go through the year, and our enthusiasm for taking on a healthier diet decreases, only to be peaked again the following year as we feel that we need to be healthier in our consumption at the beginning of the year. So that's for individual protein, but what about the way that the food is perceived as perhaps natural or processed? So here is another Google Trend search, this time a global trend, looking at the term natural food versus a more recent term, ultra processed food. So many people are now worried about the level of processing in food and whether that links to problems associated with non-communicable diseases. And you can see that the number of people searching for natural foods has gradually waned over time. And conversely, in the last year or two, the number of searches of ultra-processed food has picked up as more people are being concerned. And you can break this down by country and look to see where these areas of interest are. But it's interesting to see what these kind of trends are causing and what's underlying it, indeed, if there's anything underlying it. So how can food impact health? We know that it does, but we don't really understand all of the ways that it can help, and we need to do that if we want to improve health. So really what we want to know is how we get from this kind of spread of meals to this kind of outcome where everybody is healthy and doing exercise, as opposed to this where they will have early mortality. And it's understanding the way that food processing and food structure affects risk factors for disease, that is going to help us in that search. So I thought it might be helpful to say a little bit initially about food structure and nutrient release, so digestion. Now, you could argue that digestion starts actually before you've even consumed the food, but this is really more Louise's area of expertise than mine. So cognitive and sensory enhancement of satiety, and not only satiety, but enzyme secretion, if you think about Pavlovian responses, is far more of a psychological effect than a physiological effect. So I'll leave Louise to say anything about that. So let's think about where processing of the food and digestion actually starts, which is probably in the mouth. So with aroma and taste and texture sensing, and all of these are important as gatekeepers for quality of the food and allow us to assess whether food might be good for us or potentially dangerous, because the mouth is the last opportunity to spit out the food if it's not good for you. And also it's the place where we first start really breaking down the food, mostly through chewing, although there is also some hydrolysis of starch. And then we go from the mouth into the stomach, and you can see the stomach here, where the food undergoes proteolysis, so breakdown of proteins and lipids, and obviously acidification. But actually that's relatively limited. So mostly the stomach is a storage device, which is why we have a sense of fullness. And so we can perceive how full that is. And only towards the bottom of the stomach, in the antral region, do we have much mixing and physical processing of food. From the stomach, food passes into the intestine, and that is where the majority of digestion takes place through proteolysis and lipolysis and amylolysis, and mixing, and obviously absorption. So this is where we're targeting in some senses, because this is the first site of absorption of compounds. What you may not be aware of is that we're also sensing the nutrients going through into the different parts of the GI tract. So we're very aware that we can taste food in the mouth, but we have similar related receptors all the way through the GI tract, and they help control the passage of food through and the rates of secretions of different components. So, and they do that through a range of gastrointestinal hormones that affect things like gastrointestinal motility, enzyme and bile secretion, but also appetite. So they're key for understanding that. So I said that the stomach is essentially a storage device. So what we're really interested to know is what factors affect the rate of emptying from the stomach. And we know that there are essentially two main factors. Well, one is caloric density, where high density tends to empty more slowly, and low caloric density empties more quickly. So that's about the energy density. And also the structure of the gastric content, known as chyme, is important. So the more solid the structure, the more slowly it will tend to empty, and the more liquid, the faster it will tend to empty. And there is a sieving effect. So particles larger than about 1 to 2 millimeters tend to be retained. If you don't believe me, here is a couple of examples. So these are MRI images from a study we did a while ago now, where we compared a low energy density meal with a high energy density meal. We took images from MRI and measured the gastric volume and tracked that as a function of time. And you can see there were significant differences between the two. So the low energy density meal, these had the same energy. It was only the energy density that varied. So obviously the volume was much larger in the low energy density. Both of them emptied around about 2 kilocalories per minute, but you can see they were very different in volume. And obviously the fact that they're different in volume meant that there were significant differences in the feeling of fullness, as measured by these subjective VAS scores. And if you feel fuller, you tend to feel less hungry. So there are also differences in the hunger rating. So it affects appetite, as you would expect. If you feel full, you don't want to eat a lot. So it really, really is important to understand the calorie density and also the structure. And in terms of structure, that can be at a number of different scales. And perhaps one of the key areas where structure is important is in protein digestion. We started by looking at people's using Google, looking up protein. And here is one of the reasons why. Because different types of protein in milk are, for example, digested very differently. So, for example, whey proteins are digested quickly. So this is plasma leucine concentrations representing the amount of amino acids that are appearing in the blood after consumption of milk proteins. And for whey proteins, they appear in the blood very quickly at relatively high levels, whereas the other components in milk, caseins, appear much more gradually. And this is why whey protein has been introduced as a sport nutrition. Of course, now everybody expects that if you just consume whey protein, you'll automatically become fitter without having to do the exercise. I'm sorry to disappoint you. If you consume the protein without doing the exercise, it won't do you any good at all. You have to have the combination. We can look at that in a little more detail with very simple digestion. So here is an example again of the whey protein and the casein. So whey proteins at the top here through a gastric phase of assimilated digestion and intestinal phase. And interestingly, the whey proteins are almost resistant to digestion by the enzymes, the proteases in the stomach. So even though we've seen in the blood they appear very quickly, this is not a result of digestion in the stomach. This is a result of digestion in the small intestine. So you can see in the intestinal phase, instantly, practically, the protein is digested. So there's no intact protein left. Whereas the casein takes a little longer in the stomach, but is digested in the stomach. Again, because it's digested in the stomach, you see nothing in the intestine. Now, that doesn't match with what we've just seen in the blood. So why is that? Well, I'll come on to that in a moment, but just as a comparison with these proteins, which you can see are highly digested, here are two proteins from egg. This is lysozyme and ovalbumin, and you can see that in the intestinal phase of digestion, one of the models shown here shows there's no intact protein, but the others have lots of intact protein, and that's true of both of these proteins. So egg proteins are much more difficult to digest than milk proteins. And this is also true of a number of plant proteins. So they're much more resistant to digestion, so digest more slowly. So if you want to have that sport nutrition effect with the appearance of amino acids in the blood really quickly, that's a problem. Now, back to the milk. Why did the casein look more slow? Well, here I've got a set of videos of someone drinking milk, and these are videos from a pill cam, and the pill cam was swallowed and rests now in the stomach. The stomach is empty. How do I know that? Well, you can see all the folds here of the stomach wall because it's contracted really small and has not got much in it. So if I now play this video, which hopefully will run, or maybe it won't. Let me just turn that off. There we go. So you can see it empty, and then the happy participant drinks the milk, and we see the milk appear in the stomach, and you think to yourself, well, okay, now we have a really boring video for the next 20 minutes as it shows just white. So let's go to the next slide. And the first thing that you can see is, rather weirdly, for having consumed a liquid after 80 minutes, on the left-hand side, we see the stomach wall again, and on the right-hand side, we see the white, but there's apparently a sharp boundary between the two. And you wouldn't expect to see that in a liquid. And of course, that's because you're not seeing it in a liquid, you're seeing it in a solid. The milk has turned into a solid. And that becomes even more apparent when we run the video. And what is happening is the gradual acidification and the partial hydrolysis of the proteins in the milk is exactly the same process that you would use for making cheese. And essentially what you've done is you've made mozzarella. And that mozzarella persists in the stomach for quite some time. So here is after 160 minutes sitting in the stomach, and you can still see there's some pretty large chunks of, well, for want of a better word, mozzarella sitting in the stomach. So just because you started with a liquid doesn't mean that you end up with a liquid. And this is the reason why caseins are slow to digest, because it's the caseins that form cheese. So whey used to be a byproduct, and the same in the stomach. So it's the whey proteins that form this solid curd in the stomach. We can use this knowledge to look at meals and understand whether we can control this in a bit more detail. So this is some data from a study that we did a while ago, again using a combination of MRI and in vitro, where we took essentially the same meal in terms of calorie content, so protein, carbohydrate, lipid, but they had different structures. So one was a semi-solid structure and the other was just a liquid. We looked at the gastric behavior, both in vitro and in vivo. So you can see on the left, let me just turn my pointer on again. Here, the semi-solid system, and you can see on the right here the liquid system. And in the stomach, in the liquid system, you can see a sharp boundary, and that is where the cream layer in this emulsion has gone to the top. And you can see exactly the same thing in our in vitro simulation. And on the left, you can see the semi-solid, and you can see the boluses persisting in the stomach, and they sink to the bottom. And the same thing happens in our in vitro system. And we can use this to understand what nutrients are being released and when, and it's clear that you see release of nutrients at different phases, so either early or late, depending on whether you're looking at the semi-solid or the liquid system. And this will have an impact on fullness, for example, but also the appearance of these amino acids in the blood. So by controlling the structure, we can control the either sedimentation or creaming in the stomach, and the rates of emptying. And we can do that to a certain extent by using different types of processing. So here, for example, we've used milk, either raw milk or ultra high temperature processed milk, or homogenized milk or homogenized and ultra high temperature process. And although it's not terribly clear to see in these, you do have sedimentation with the raw and ultra high temperature process. And you see creaming, i.e. floating to the top as opposed to the bottom in the UHT and homogenized. So just from using different processing, you can control how milk behaves when you consume it. There are other factors that control how the food behaves in the stomach. So one of the things that we've done more recently is to go back and look at some of this data. So in the images that you see here, so I should explain perhaps what you're really looking at. So this is a participant lying on their back. This is the spine. This is the stomach, so which is mostly in white, and the liver is on the left. So in these sequences, liquid So water is shown up as white, and low water areas are shown up as dark. And you can see these dark regions, which are boluses of the semi-solid material that were formed in the mouth and then swallowed, and they stay in the stomach for quite some time, and we can measure that. So we can measure all of these and convert the boluses into a size distribution. Not only that, we can then total up the volume of the bolus and see how that bolus volume changes with time and compare that with rates of overall emptying. So in this study, we were able to measure the emptying rate of liquid from the stomach, liquid and semi-solid from the stomach. Obviously, the semi-solid material tends to stay behind, and that is the boluses that we're looking at. And one thing that we found was that the emptying rate only varied in these meals really in the first kind of 60 minutes. After that, they were virtually identical emptying rates. So something varies between the two meals in that early period. And the main thing that varied is this bolus volume, because after that, the boluses kind of disappear. This is for our particular meal, but you can do the same sorts of things for more conventional meals. So this is some data from colleagues at the University of Nottingham after a particular meal, and we've just published something looking at bolus size as a function of meal volume. And I think that's something that we'll continue to look at, because the persistence of this structure in the stomach affects appetite and emptying. Other things also affect it, so things like cellular encapsulation. So if you eat a nut, for example, an almond, you don't break open every single cell, and the cells that don't break open tend to hang on to their nutrients. Here's the same sort of thing for carrot. So this is what a raw carrot looks like. You can see the carotenoids orange in the center of the cells. And then these are debris from ileostomy patients. So these have been through the small intestine of a human and then been extracted at the end of the small intestine, and you can still see the orange carotenoids sitting in the cells, even though they passed all the way through the small intestine. So if you don't break open the cells, you don't extract the nutrients. So raw carrot may sound like a great idea, but actually not as good for you as cooked carrot, because you don't break open every single, unless of course you can chew really, really finely, but that's kind of challenging. So structure at different levels and in different types of structures is important. Here's another example with the almonds. So in this case, the starch remains behind. So the purple areas are starch in the cells. And you can see here it's really still intact. And actually the figure on the left, there is starch in this light region here, but the cells are so intact that the stain was unable to penetrate into those cells and stain them up. So that's why they were still white. So the cells can hang on to nutrients unless you break them open, either by cooking or by some other mechanism such as chewing. Just a quick one here that I put in for Louise's benefit, looking at ferulic acid and its ability to block enzymes. So there are some phenolic structures that are able to impact digestion because they can block specific enzymes or specific transporters. It's unclear how important this effect is in real foods, but it is certainly something that we're starting to look at. So I'm aware of time, and so in conclusion, I hope this whistle-stop tour has been of interest and highlights that both bioaccessibility and bioavailability are a function of digestion kinetics and can affect things like metabolism. Factors affecting the digestion kinetics are a function of structure at lots of different scales. And these are structures that are formed both before consumption and after consumption. So just because a food doesn't have a structure like milk before you consume it doesn't mean that structures formed subsequently aren't important. And we've seen that both endogenous and exogenous enzymes and in particular physical processing are involved in these processes. So choosing the right food structure may end up being a matter of life and death in the long run because of these risk factors. I hope that was of interest to people, and I'm happy to answer any questions. So thanks very much. Here's some acknowledgements for all the people who helped out in these studies, and I'll leave you with the reminder about the Q&A in the chat.
Thanks very much, Alan. I don't know whether you've put me off milk or mozzarella. I'm hoping people will have posted some things in the chat. At the moment, it's more complimentary stuff about your talks, but which I did think was really great. And I thought this was a really difficult topic made really accessible to lots of people. And that seems to be reflected in the chat. I've got a question though, and it was something that I was thinking about as well, Grace. And the question is, would the processing in plant milk or plant-based dairy have the same effect in terms of digestion and separation? And I was thinking of it more simply because you showed almonds, and people are now making lots of things like almond milk. Can we answer Grace's question?
Plant-based milks are not really milk in the conventional sense. The only similarity is that they're white. They have very different protein content and protein profile. They have a different lipid content. They also have different mineral content. So it made me laugh the other day when I was talking to my sister-in-law, and she told me she'd started drinking soy milk because it had added calcium, and she felt that the calcium was a benefit. But obviously you have more bioavailable calcium in cow's milk than you do in soy milk, so you need to be a little bit careful about that. So I think the short answer to that question is no, they don't behave in the same way because they're very different products.
Okay. So I think Christine's asking a similar question, which is about plant milk proteins versus bovine proteins.
So plant proteins tend to be A, less soluble, and B, less digestible than some animal proteins. So dairy is particularly good, but of course dairy is good largely because there's been millions of years of evolution to perfect the formulation and provide nutrition for infants, whereas plant proteins are there to usually as storage proteins, so they're there to provide a long-term source of energy for a plant, and that works in a very different way. So solid, you don't want something that's highly soluble, so you can imagine something that's insoluble and locked into a cell is much more useful to a plant, but not so useful for us.
Okay, and then we've got another question from an expert. So Brent Murray has asked, presumably the raw milk settles out because the fat gets trapped into larger protein structures and aggregates?
Yeah, the aggregates sufficiently outweigh the buoyancy of the fat in raw milk, and I think as he says, it's likely that you get some phase separation of lipid at the top, but the bulk sediments. Whereas if you homogenize the milk, which went the other way, it's much more finely distributed and doesn't separate out as quickly.
Excellent. And I think what we will do with your talk, Alan, is we'll put up a list of publications, because a couple of people have asked about the digestive behaviour video and if that's published. So if we can do that, that would be helpful. And then another question from Henry Greathead. Are intact cells that pass through the small intestine important for the health of the hindgut? They provide nutrition to the microbiota, ? And therefore, should we aim to not fully break down the cell structure in the mouth, stomach and small intestine? So it's about passage through the gut here, isn't it?
I quite agree, and this is about having a balance of soluble and insoluble fibre. So obviously these cells represent largely insoluble fibre, and it's important to have insoluble fibre as a component in the diet as well. So yeah, absolutely, they have benefit in the hindgut.
Excellent. And then if we think about the bioavailability of raw carrot, would this be the same for other raw veg and fruits, or is it more to do with the carotenoids?
Partly it depends on the strength of the cell wall. So for sure it is true of other veg and some fruit. Particularly ripe fruit tends to have quite soft cell walls, although if you think about things like chickpeas, they're a problem. If you think about mealy apples, the reason that you have that mealy texture is because the cells are starting to break apart. So they might well pass down the gut, not terribly well broken up.
Okay, I think we've got another question about the difference in processing between cow's milk and plant milk. But I think there are variations in processing of cow's milk too. There
are, yeah. So virtually all cow's milk that's consumed in this country has been thermally processed. So it's been heated, it's been pasteurized. It's difficult to get hold of raw milk that hasn't. A lot of it has also been homogenized because it breaks up the droplet size. So although people like to think that they would like to have minimally processed milk, actually that's not what people buy. And generally people don't like their milk to separate out and have a kind of layer of cream on the top. I don't know why, but homogenization prevents that. When I was a kid, you used to take the cream off the top of the milk.
Yeah, you're bringing back memories. And I suppose my question with that is, people are buying ultra filtered milk, so you know the premium price. I don't want to say a name, but the word Cravendale comes to mind.
Indeed.
You know, what's the difference? Would it make a difference?
It does make a difference. So they really putting this stuff through ultrafine filters in order to remove any bugs, bacteria that might make it go off. So this is why it has really long life, because it's processed in a different way without necessarily being thermally processed. And different processes will have different end points in terms of the types of behavior through the gastrointestinal tract, as obviously before you get to that as well. So okay. A lot of processing is designed to affect shelf life, because the longer the shelf life, broadly, the easier it is to make sure it's still in a good condition when the consumer buys it.
And it tends to last longer than in the fridge as well.
Indeed, yeah.
But the processing by the gut would also be different.
It would.
Okay. So another question from someone whose name I can't pronounce, Shia. You mentioned that casein digests slower than whey protein.
Yeah.
Based on the fact that casein being present in milk has casein micelles. Are there any differences between the digestibility of native casein micelles, acid casein, and rennet casein?
Well, there are some differences. So in milk, generally the caseins are in the form of these micelles, which are fairly large aggregates of protein, and they're there naturally, and they're part of the way that the milk delivers calcium to the body in the form of calcium phosphate. And when the casein is treated in other ways, these micelles tend to fall apart. So especially for things like renneting, you not only pull the micelles apart, but you start to hydrolyze the casein itself. So you only tend to see these processes in the intact micellar casein, and the effects get less and less the more you process the casein away from that.
Interesting. So I don't think there are any more questions. Lots of positive comments, Alan. I think I reiterate those. Thank you very much for a really excellent talk. I think you've done milk this week. I think we'll get you back to do some other nutrients, some other foods, and show us some more of those really exciting. Thank you very, very much.
You're very welcome.
And for everybody who's still on the call, the video of this will be available on the GFEI website, and Alan will add some papers to it as well.
Yep.
Thank you very much, everyone.